Amit is a team lead at Sense Research Foundation. He brings together two exciting fields of research together, which one is the impact of immune senescence and also in general senescence and how it impacts aging. Over to Amit.
I am Amit Sharma from Science Research Foundation. Pardon. I am from Strategies for Engineered Negligible Senescence Research Foundation. And we exist mainly for one big reason, which is the silver tsunami that we were all hearing about is here. So by this year, 1 in every 7 individual in the US is over the age of 70. And with this increasing age, there is increased susceptibility to many diseases of aging in all developed world. So a better understanding of how to circumvent this will allow us to develop novel therapeutic interventions. So we know that aging accumulates in our body because of various kinds of damages that start building up simply because we age. Is a matter of time. There are more protein aggregates, both intracellularly, extracellularly. There are more mutations in the mitochondria, and mitochondrial dysfunction starts happening. The telomeres start become shorter, and there is more DNA damage foci or in all of the cells. We have an accumulation of senescent cells, and the matrix, the extracellular matrix starts becoming stiff, so the organ starts malfunctioning, and the stem cells don't behave as well. The numbers are lost, or they don't really effectively do any regeneration function, and the immune senescence occurs. All of these damages are fixable, as Aubrey talked about before. So when all of these happen over the time, we start getting old. My lab mainly focuses on 3 of those interventions. So what are senescent cells? Like the most simplistic, basic definition of senescent cells is that any cell in our body which is undergoing DNA damage tends to accumulate. We try and fix those damage. The DNA we cannot necessarily repair all the time, and the telomeres become short, and that also contributes to DNA damage. This mechanism essentially is been considered as a way, like a way by which a cell prevents from becoming a cancer cell. So when there is an activation of oncogene, again, cells become senescent. From a definition, the way we understood this before, all of these changes contribute to certain kind of phenotype in the cell, which is DNA damage leading to senescence-associated heterochromatin foci, the actual DNA changes that are happening, or epigenetic changes that are happening to the DNA, the increase in P16, P21, and increase in lysosomal dysfunction. Now, yeah, that is very simplistic, and yeah, we've been trying to target senescent cells based on all of these phenotype, and mainly because they resist cell death, so they increase these pro-survival pathways. So Andrea's not here, but he was talking about how senolytics don't work in senescent macrophages, and we'll get to that. But I think it is because of what senolytics. So we also found that we recently published a paper that not only these senescent cells differ based on the type of damage, they also differ in another way, meaning like if they're not exposed to any of these damages, but they're exposed to just the inflammatory proteins chronically, they become senescent in some ways, but they're different in another. One thing is pretty obvious, that they are involved in lots of diseases. Pretty well established now that senescence accumulation contributes to many diseases in our body. And we have ways to get rid of them. The immune system is very tightly regulating senescence burden in our body, so there's a lot of redundancy in the system. But we age, senescent cells accumulate, so what is going on? There were 2 terms that have been thrown around a little bit around here in this conference, immunosenescence and inflammaging. What really are these things? So when we talk about immunosenescence, we're talking about the decline in how hematopoietic stem cells behave. The thymus disappears. The pool of naive T cells and B cells decline quite significantly. We have T cells, cytotoxic T cells in much larger numbers. but they're not functional. The diversity of TCR and antibodies also significantly change. And all of these things add to our susceptibility to infections, cancer, the vaccines don't work as well, and we develop autoimmune diseases. Inflammaging is essentially chronic inflammation that builds up because we accumulate a lot of pro-inflammatory cytokines and chemokines that again increases our susceptibility to lots of diseases and disorders. So what happens first? Is it the immune system that becomes dysfunctional and then is not able to get rid of senescent cells and they start building up in our body? Or is it that there are so many more senescent cells that immune system just cannot keep up anymore, and we don't really quite understand yet what is the cause and what is the effect? A few things we know. For instance, we know that there is a myeloid bias. We make more monocytes and macrophages as we age, and we also know that the number of immune cells changed quite significantly. So there is this research that came out from Zurich where they characterize immune cell composition in various tissue in our body as we age. They just looked at young and old mice, and they were able to see some takeaway messages were from that study was that the major population that changed within the tissue is NK cell, CD4 T cells, and we see a significant change in plasmacytoid dendritic cells. And that can explain why we are more susceptible to viral infections as we age and why we have less response to vaccines. Now, hematopoietic stem cell is another interesting population. It makes all of the blood cells in our body, and they are quite affected by aging. Aubrey talked about that. And one of the common themes that I want to draw attention in the next few slides is that these damages are consistent, and we have to really now rethink our definition of senescent cells. So not all senescent cells are the same, and there are different kinds of senescent cells. So for instance, hematopoietic stem cells tend to accumulate mitochondrial damage. They accumulate persistent DNA damage foci. Their telomeres are much shorter, and they have proteotoxic stress and epigenetic changes, which reduce their ability to self-renew and contribute to this myeloid bias that we see with MSCs. There are hopeful therapies. For instance, we know that the bone marrow transplantation from young mice has a value. So young mice bone marrow tends to behave much better than older ones, and transplantation seems to not only improve cognitive function, memory, but also extends lifespan quite significantly. And then there are these interventions that people have talked about. For instance, CDC42 is one of the factors that seems to be elevated in older HSCs, and its inhibitor, Casein-9, seems to improve their function, their ability to differentiate into the cells we like, and p38 MAP kinase inhibitor seems to be also very, very effective. Thymus, again, an organ that breaks down as we age, and there are therapeutic interventions. People have talked about adoptive transfer, organoid transplantation, and they're also talked about gene therapy approaches by overexpressing FOXN1 in fibroblasts and then engrafting them and showing how they can restore thymic function along with various kinds of chemokines and cytokines. One of the cell type that we like to work with is T cells, and the increasing incidences of DNA damage and production of pro-inflammatory cytokines is well known. We know that these T cell diversity is changed with age. We know they express the co-stimulatory molecules at much lower levels, especially in T cells that have accumulated when they have shorter telomeres. And they express CD57 a lot more, and they start behaving more like NK cells with age. This mechanism is because of mitochondrial dysfunction that accumulates in T cells. And this mitochondrial dysfunction and change in nutrient sensing pathways is regulated by a protein called cystrin. You can manipulate that and improve their function quite significantly. And similar changes. So, based on that, there are various engineering approaches that are out there that people are looking at. One approach that we are interested in CAR, and I'll get to that in a second. Again, same changes in B cells. We know they have much shorter telomeres. They become damaged with age, and the B cell diversity also changes. We have an accumulation of aged B cells, adipose-associated B cells, which are phenotypically different, but they are also kind of senescent-like because they lose their functionality and they become very inflammatory. Again, if Andre was here, we would have talked about this. I mean, I agree with him that a lot of macrophages that accumulate in our body are actually senescent-like, especially with age. They don't produce as much TLRs, they don't express as MHC, they're not really good at presenting the antigen, and they are contributing significantly to inflammation. There are approaches that people are using, but they're also senescent-like. And all of these types of senescent cells may or may not respond to conventional senolytic drugs. So what is the answer? My personal favorite answer is natural killer cells. So they also undergo a lot of phenotypic and functional changes with age. Their population changes. There is a balance between NK cells that express high levels of CD56 versus low levels of CD56 and cooperate really well. That changes with age, and they don't respond to IL-2, so they don't proliferate as well with age, and their effector function also declines. And in my lab, we are doing 3 different kinds of interventions to improve the NK cell function, and those are the 3 approaches we are trying. They are very important in regulating senescent cells. In fact, the main homing signal that is regulated by the master regulator of senescence, P53, and messing with their effector function can also cause significant increase in senescence burden and decline in lifespan in mice. This mechanism by which they regulate senescence is also well understood. So we know that senescent cells express eat me and don't eat me signal, and NK cells are very good in recognizing them and then causing cytotoxicity and removing senescent cells, and senescent cells have found ways to escape the immune function of NK cells. So there is a limit to what we can do with them. So when we were trying to understand the diversity of NK cells within blood, we found that the NK cell numbers quite significantly changes in mice as well as in humans. The numbers change, and their ability to proliferate to infection also significantly changes. They are cytotoxic, we know that, but the cytotoxic function is not perfect. We were thinking of how to improve them. So there are ways we are working in the lab to rejuvenate them using a cocktail of cytokines and chemokines. And we are also trying to engineer them to improve their cytotoxic function. And the work that I'll discuss today was done by these 2 very talented people in the lab. So we first went back and looked at the surface of senescent cell, and we were only focused or interested on the proteins that were expressed outside of the cell and then can become targetable. So by pulling them off the membrane of senescent cell, we did proteomics, and we identified 22 proteins that were conserved in 2 different modes of senescence induction. So what is a good target? To identify a targetable marker, it has to be highly expressed on senescent cells. We're talking about cells that are behaving differently, so we won't expect to have a new antigen. It would be something that may be present on normal cells, but it has to be high enough in senescent cells that we could target them just based on that. And it should be present in multiple models of senescence, not just one, because they're all very different. And it should be expressed in all kinds of tissue when they accumulate senescent cells, and they should be amenable for therapy. So based on that, we identified several targets. One of them that we were interested in is here, SRF5, and we confirmed that by different models here. So this is one such model where we found that SRF5 was elevated in fibroblasts that were induced with doxorubicin to induce senescence. And we found a similar increase in the expression of this protein on the surface of multiple models of senescence. This is replicative senescence and also etoposide. And since then, we've also confirmed its expression in endothelial cells along with fibroblasts. Now, the question is, can we target senescent cells based on its expression of SRF5? So we engineered our CARs where we use 3 different antibodies and clone the SEFEs in our standard CAR construct. So we used a CD28 structure, the hinge, the transmembrane domain, and the intracellular domain. And we changed the length of the hinge because that allows it to get the flexibility to be able to identify the target and bind. Unlike an antibody, which is pretty flexible on itself, when we bind it to a surface of a cell, then we really need a little bit more flexibility. So when we did that, we confirmed that this CAR was functional. It's highly expressed on our NK cells, or NK92 cells. We confirmed that. And then we performed our cytotoxic assay, where we used this approach where we could monitor the viability of cell based on its ability to disrupt the flow of current. So there are more cells, there is more resistance, less cells, less resistance, fewer cells. And that will allow us to actually monitor it over a period of time and basically a real-time analysis of cytotoxicity. So we found that our CAR NK that was able to target senescent cells based on the expression of SRF5 was significantly more toxic to senescent cells in a shorter period of time. The other thing we found is that if we used an antibody to neutralize the natural receptors that NK cells recognize, which is NKG2D, we were able to see a significant decline in the cytotoxic function of NK cells, whereas these NK cells that were engineered were actually not inhibited in their ability to find and kill senescent cells. And we have done this many times now, so we're quite confident this works. Now the question is, where are we going from here? Our long-term plan, or near-term plan, really is to see if we can engineer cord blood or NK cells derived from a healthy person and engineer them with a viral vector, and then make off-the-shelf CAR NK, and then try to treat multiple diseases. The disease that we want to go after now first is IPF. And hopefully we will be able to then show that CAR therapy based on CAR NK therapy could be applicable for diseases other than cancer. And that's where we are at for now. This work was done with my very small but nimble team. All of this work was done while the world was shut down by COVID. So I'm very proud of these people who showed up for work every day and continue to work with us. I'm also thankful to the donors, Vita Dow, who supported this work, USNAM, who've given us resources, Forever Healthy Foundation, and more importantly, donors at Sense Foundation, who's given us money to do this work. Most of you probably know Sense through Aubrey and the kind of work we've been doing. The idea is that we try and do stuff in-house and stuff outside, fund research that is able to target those interventions that we all had heard Aubrey talk about earlier, and they've been reasonable success. So if you have a project idea where you think you have an intervention that can be used to target senescence or other types of damages that accumulate, please reach out. If you don't want our money, we'll take yours, give us donations so we can do some of our work in-house. We also have an education program where we try to train students at different levels, the next batch of researchers who can continue this fight against aging. With that, I'm done.